So I'd like to welcome everyone to our monthly Tennessee Center for AIDS Research seminar series. This is a internal and external seminar series that's available nationally and internationally, where we highlight leading researchers in the HIV field from a multitude of different disciplines for 1 hour of presentation and discussion. So today we'll be hearing from Dr. Chandravanu, or CV Dash, who's a good friend of mine, as well as being a tenured professor and internationally recognized virology researcher on the faculty at Meharry Medical College. And Dr. Dash received his PhD from the National Chemical Laboratory in India, followed by a postdoctoral training in the HIV drug resistance program at the National Cancer Institute, part of NIH. And then in 2009, he was recruited to Meharry with a highly competitive NIH K99/R00 Career Development Award. And he currently serves as the chair of the Department of Microbiology, Immunology, and Physiology at Meharry Medical College, as well as the executive director of the Center for AIDS Health Disparities Research at the same institution. Dr. Dash has over 25 years of experience in the biomedical research field and has made seminal contributions to the field of HIV, primarily in the area of virology, with more than 65 peer-reviewed articles. He has been continuously supported throughout his career by the NIH, and currently he is the principal investigator of no fewer than 3 R01 grants. And he's also the consortium PI on another R01 subaward. And importantly, he also has the rare distinction of being the only K99/R00 fellow among approximately 3,000 of these awards, who has successfully transitioned to R01 funding at a HBCU medical school. Currently, Dr. Dash also serves as the director of the developmental core of the Tennessee Center for AIDS Research, and he and I worked together on in that core for many years. And he's also the co-director of the NIMHD U54 funded Research Centers in Minority Institutions Program developmental core at Meharry. He leads the effort on the NIH R25 Enhancing Virology Training, or NVEAT, program for minority students. And he's also the lead for the Tennessee CFAR training program on Diversity, Equity, and Inclusion Pathway Initiative. And he was recently awarded the 2021 Basic Science Researcher of the Year, as well as a number of other awards. And lastly, in addition to all of his scientific accomplishments, Dr. Dash has also been very passionate about training the next generation of scientists. He has a long history of mentoring, and his laboratory is dedicated to strengthening the resources and career development opportunities for minority graduate students and postdoctoral fellows. To date, he has graduated 7 PhD students. He has trained more than 35 undergraduate students and several postdoctoral fellows. And importantly, many of his trainees have successfully transitioned to careers in academia, industry, and other sectors. So please welcome Dr. Dash. Thank you, CV, for being here. He'll be presenting the Tennessee Center for AIDS Research Seminar, Nuclear Role of HIV-1 Capsid. Take it away.
John, thank you so much for that wonderful introduction. So it's a real privilege to tell you about our current research on HIV-1 capsid. Can you hear me clearly, John?
Yeah.
Okay, I'm going to take away my camera so to be a little bit easier. As indicated, I am at the Meharry Medical College in Nashville, Tennessee, which is part of the Tennessee CFAR. It's a multi-institution CFAR involving Vanderbilt, Meharry, Tennessee Department of Health, and Nashville Cares. So let me see if this works. John, it forwarded, right? The slides went ahead?
Yep.
Okay. So I do not have to give this introduction, but I thought this is very important. That HIV-1 is an enveloped RNA virus that infects CD4 T lymphocytes. Here is an electron micrograph of the virus, which has been taken. And here is a schematic representation where you can see that the virus, in this case, the particle HIV-1, has a membrane surrounded by the envelope proteins. And inside, you see a conical capsid that contains the RNA genome, 2 copies, and the necessary viral and host factors that are needed for productive infection. And as we all know, chronic HIV infection causes one of the deadliest diseases called as acquired immunodeficiency syndrome. And it is important for us to get reminded that HIV/AIDS continues to be a global epidemic. For example, if you look at the data, there are almost more than 80 million people who have been infected with the virus, out of which almost 40 million people have died. And currently, greater than 40 million people are living with this virus. And a little more details in 2023, there were almost 1.3 million new infections in the world, and there are almost 630,000 deaths in the same year. The point here is we have a global epidemic that we cannot take our eyes off, and we need to develop strategies to reduce the impact of this public health epidemic. Now, fortunately, there are a number of potent antiretroviral drugs that are available in the market that effectively control HIV/AIDS. And to this crowd, I do not have to go through the medicines available, but essentially, there are 7 different classes of drugs that are available for HIV treatment. But as with any RNA viruses, for HIV/AIDS, ART is not curative. Given the RNA nature of this virus, HIV develops drug resistance to almost all drugs. Many of these drugs continues to be toxic, and there is comorbid conditions associated with HIV-infected individuals who are virus is under control. So the point here is we have to continuously identify new therapeutic targets to effectively control HIV/AIDS. And as many of you know, a couple of years ago, there is a new drug that has been making a lot of splashes. It is developed by Gilead Sciences, and it is a drug that targets the HIV-1 capsid protein. In fact, this inhibitor or this drug is so potent that it is being considered to be used for long-acting injectables. And the drug's name is lenacapavir. And I think most of you know, in September, one of the studies that came out, the use of lenacapavir for HIV prevention, in fact, it was so effective that Gilead Sciences stopped the blinded phase trial and decided to offer open label this capsid inhibitor to all participants for prevention. And this provides you a glimpse how basic science studies inform to develop better and highly potent drugs that could transform the course of HIV disease. But to basic science researchers, HIV capsid has been an attractive therapeutic target. It's mainly because this protein, which is now onwards I'll say CA, is genetically fragile. What does that mean is if there are any mutations in the capsid protein, then chances are very high that the virus loses its infectivity. For example, the fragility of capsid is 70% compared to integrase, which is 13%. And just to remind you, integrase inhibitors are one of the best inhibitor used in the clinic currently. So the point here is we know that HIV-1 capsid is an attractive therapeutic target. However, the mechanisms by which HIV capsid regulates virus infection has not been fully understood. And that's what I will tell you a little bit about from our efforts in the laboratory. Now, what is HIV capsid? It is the building block of the viral capsid. For example, I showed you a schematic, and here is the viral particle, and here is the capsid that is conical in nature. But if you look a little more deeply, the capsid core contains hexamers of the capsid protein and pentamers. There are 12 pentamers, 7 on the top, 5 on the bottom, and almost 200 hexamers in this capsid core that are made from, as I indicated, hexameric and pentameric structures of a single capsid protein. That means this capsid protein, which is P24, 24 kilodalton protein, organizes in a beautiful manner to give rise to hexamers and pentamers to form a capsid core that is critical for viral infection. And just to remind, this capsid protein is encoded in the HIV-1 gag polyprotein, which contains matrix capsid, nucleocapsid P6, and spacer proteins. So traditionally, we know that viral capsids are known to protect and deliver the viral genome to the target cell. And they come in different flavors. They can be icosahedral, helical, or mixed. And in case of HIV, it comes in a conical shape. And what we have been learning in the last 20 years is capsid is not just a container or box. In fact, it promotes a number of different steps of HIV infection in a target cell. And just to quickly remind you, this is a schematic of HIV replication, which is highly complex. Once the virus binds to the target cell through the CD4 and CXCR4 or CCR5 co-receptors, the capsid is released into the cytoplasm, and the RNA inside the capsid starts to reverse transcribe into a DNA copy. And once the reverse transcription is completed, the viral replication complex is called as pre-integration complex. And this pre-integration complex carries out integration of the viral DNA into the human genome. And this is a defining step because once the viral DNA is integrated into the human chromosome, it stays there for life. And thereafter, through transcription, viral genomic RNA and viral proteins are produced, and these assemble in the plasma membrane to produce virus particles that are become infectious after maturation step. But what we know so far from studies from a number of laboratories that capsid protein helps in reverse transcription. It also helps in the nuclear entry of the virus for viral DNA integration. In addition to that, the capsid provides scaffolding for a number of host factors that are essential for HIV replication. In addition, because the capsid serves as a protection, the viral DNA is protected from target host restriction because of the capsid protein as well. So it cloaks the viral DNA from cellular sensors from degradation. What I'll tell you a little bit today from our efforts that the role of capsid doesn't end there. In fact, capsid also regulates one of the fundamental steps of HIV replication called as viral DNA integration. And these studies of capsid is much more important at this moment of time because in the last 5 years, the field has really undergone a transformation in our understanding of capsid's role. For example, traditionally it was believed that once the virus enters the cell and reverse transcription is completed in the cytoplasm, the capsid falls down in the cytoplasm, followed by the pre-integration complex going to the nucleus for integration. So uncoating believed to be occurring in this uncoating of the CA in the cytoplasm, then reverse transcription is completed in the cytoplasm, then the PIC goes into the nucleus for integration. This model has been challenged severely with recent developments from a number of labs. What quickly I want to tell you, apparently, the current model is an operationally intact capsid is able to enter the nucleus, and the reverse transcription process, which is believed to be previously completed in the cytoplasm, gets completed in the nucleus, and then uncoating occurs uncoating of the capsid occurs immediately before the integration of viral DNA into the chromosomes. So this fundamentally changes our view or our understanding of the role of capsid in during HIV infection. And it also should tell us how complex capsid's role has been in HIV infection and why it has taken so long for us to develop drugs against this critical viral factor. So what role does CA play in viral DNA integration? It is not easy to study them because of its multifunctional role, but we're taking a very multidisciplinary approach to study capsid's role. We are using antiviral compounds. We're using genetic approach to use specific CA mutants. We are also studying capsid binding host factors to apply cellular biology approach. And finally, we have been utilizing the pre-integration complex for biochemical studies to better understand capsid's role in HIV DNA integration. And I'll quickly tell you how these approaches in combination have been hugely useful for us to understand CA's role in HIV infection. So quickly, as I indicated, capsid inhibitors are being used for specific purposes. In fact, these are ultra potent. And these inhibitor efforts have been going on for many years, and there are a number of inhibitors that have been available to the researchers to study. In fact, GSCA or GSCA-1 is one of the precursors of lenacapavir. And before GSCA-1, it was PF74, a Pfizer compound that paved the way for the development of ultra-potent capsid proteins. And we use both PF74 and the Gilead compound to study CA's role. And quickly, here is an antiviral assay of PF74 we had conducted a few years ago. And as you can see, PF74 potently inhibits HIV infection. Infectivity is in the y-axis and the concentration of the CA inhibitor PF74 in the right, in the x-axis. And you can see in micromolar concentration, PF74 inhibits HIV infection quite robustly. But the question was, because HIV infection involves so many different steps of a replication cycle or so many different steps, we really wanted to know at what stage of HIV infection this inhibitor acts. And to do that, we can stage the exact step that is blocked by the CA inhibitor using real-time quantitative PCR assays. We can measure reverse transcription levels. We can measure how much of the virus has gone into the nucleus, and we can also measure the levels of viral DNA integration into the chromosomes. And these are done using, again, qPCR. I'm not going to go into the details of the methodology, but if anybody is interested, I'll be happy to share that after the talk. So, and here is the data. Quickly, without the inhibitor, you have infection, reverse transcription, nuclear entry, 2 markers, 2 LTR, 1 LTR, viral DNA integration. This is normalized data, and I want you to focus on the 2 micromolar PF74 effects. As you can see, infectivity is reduced dramatically, almost 95%, but this capsid inhibitor does not inhibit reverse transcription, but it does inhibit nuclear entry of the virus twofold, but this inhibition was not quantitatively similar to the infectivity defect. But if you look at the viral DNA integration inhibition or blockage, it mimics exactly how the level of defect at infectivity, suggesting perhaps for the first time that a capsid inhibitor inhibits HIV infection by blocking viral DNA integration in the nucleus. In fact, this study, which was published in 2019, preceded the study from Gilead Sciences by Ant et al., where they also demonstrated similar mechanisms of the lenacapavir original compound of lenacapavir called a GSCA1, where you can see this level of infectivity is here. You can see the 2LTR circles, this is nuclear entry is not affected, whereas integration is reduced dramatically, that is quantitatively correlated with the infectivity defect, whereas reverse transcription was not affected at this concentration. So what I'm trying to suggest here is that the studies of CA inhibitors and how their mechanisms of inhibition has been instrumental in the development of the ultrapotent capsid inhibitors you see from Gilead Sciences. Now, after the pharmacological approaches, let me give you a little bit of glimpse of how we are using CA mutants to understand how HIV-1 capsid protein is involved in viral DNA integration. There is a number of CA mutants that have been developed by Chris Aiken's lab in Vanderbilt, and these mutants have been hugely useful in understanding CA's role in HIV infection. However, we were looking at CA mutants that are clinically important. And fortunately, there are a couple of naturally occurring CA mutants which are available that we looked at. And some of these have huge importance in cytotoxic T lymphocyte mediated control of HIV infection. And these are called as CTLs. And I do not have to go through this slide to this crowd, but what we know is upon HIV infection, after a few weeks, the viral load is controlled by the immune system. And one of the critical players in the immune system is HIV-specific CTLs. As you can see, the HIV-specific CTL levels remain robust and correlated with the lower level of viral load. But after a few years, as you can see, the viral load starts to increase concomitant with the lack of the CTL's ability to kill these infected cells. So what I'm trying to say is there is a correlation how the cytotoxic T lymphocytes are unable to perhaps kill HIV-infected T cells. And one of the key epitopes that are utilized by the CTLs is the capsid protein. In fact, the capsid protein is presented by in the infected T cells, and MHC molecules recognize these epitopes to kill the infected cells. And there are a number of epitopes in the capsid, and one of the epitopes called as KK10 epitope has been fascinating for many ways, so as other epitopes. But I'll tell you the story of KK10 CA mutants here. So as you know, the CTLs will kill the T cells that are infected with the wild type virus, but the virus will introduce a couple of mutations in this epitope. These are called as RKLM. The RKLM capsid mutants, they are resistant to CTL killing, but these mutants cannot replicate effectively. After a few years, this virus acquires a complementary mutation called as SA. And what happens with this double mutant or triple mutant is quite interesting. And here is the data. Here is the wild type virus, and when we measure infectivity and we compare that to the RK mutant, these are CTL escape mutants or RKLM, you can see these two CTL escape mutants cannot replicate effectively. However, once the SA mutation comes along, now this virus is CTL escaped and can replicate very nicely. So this is a double whammy for the natural control of HIV infection. Now, when we measure HIV integration, or integration of these different viruses, what we found was very interesting. In fact, compared to the wild-type virus, the RK and RKLM viruses were hugely deficient in their ability to integrate in the human chromosome. But the RKLM-SA virus was able to integrate robustly, even though slightly lower, but significantly higher than those of RKKL and RKLM virus, suggesting that this mutant mutation, the SA mutation, restores the virus's ability to integrate into the human chromosomes. And this is not due to any defect in reverse transcription. As I showed in previously, integration is dependent on reverse transcription and nuclear entry. And because capsid is involved in both those steps, we have employed a method to measure efficiency of reverse transcription and efficiency of nuclear import. And you can see from these analysis, the integration defect is not dependent on reverse transcription or nuclear import, providing further support that clinically relevant CA mutants are deficient or blocked in HIV DNA integration. So in addition to this observation, there was another observation from our CTL CA mutant study that was very interesting. In fact, this is what led us to study cell biology approach to in our research. For example, the RK-associated integration defect was only manifested in an infected cell that expressed a host protein called as cyclophilin A. And this was reported a long time ago by two groups. In fact, in one of these studies, Chris Aiken was also an author. And cyclophilin A is known, cyclophilin A or CypA is the first host factor discovered to bind to HIV-1 capsid. And this interaction was demonstrated to promote HIV infection. And one of the mechanisms is that this interaction protects the capsid from the restriction from another protein called as TRIM5 alpha. But the interesting thing is cyclophilin A is a cytosolic protein. But this led us to ask a couple of simple questions. If cyclophilin A is involved in inhibition of integration defect of these CTL mutants, what happens if we take away cyclophilin A from the target cell? So we used T cell lines, that is parental cell line with cyclophilin A, without cyclophilin A, and then measured integration efficiency of the wild type virus and RK mutant virus. And this is what I reported, shown before. Wild type virus integrates into the human chromatin very well. RK is deficient or blocked in viral DNA integration. But if you take out CypA from these cells, what you see is quite interesting. In fact, without CypA, the wild type virus and the RK mutant virus can integrate into human chromosome almost at comparative level, providing further support that a capsid binding host factor regulates or serves as a restriction factor here for capsid mutants. So that led us to apply the cellular biology approach to study capsid's role in HIV infection. And I think I have a few more minutes to tell you a little bit more about our biochemical approach and perhaps the cell biology approach. But just to remind you, HIV infection of a target cell is critically dependent on a number of host factors. Here is a schematic. I talked about CypA. In addition to CypA, there is CPSF6, NOP153. These are nuclear proteins. And these host factors specifically engage with the capsid to promote HIV infection. And therefore, these are great tools to study capsid's role in viral DNA integration. So since CypA binds to the HIV-1 capsid, we wanted to ask does CPA play a role in HIV integration? So what we did in this case is we took Jurkat T cells, which express CPA, and then depleted of CPA. Then we infected these cells with particles. Then we isolate DNA, and we can quantify all the steps in addition to analyzing infectivity. And the data is presented here. Here is the infectivity data, and here is the proviral integration copies in infected cells. What you see is pretty clear. So CPA, lack of CPA significantly reduces infectivity. And here we have a fibrin alternative controls that inhibit HIV infection in both cell types. And if you look at the level of proviral integration in these cells, what you see is a photocopy of the reduction in infectivity and reduction in integration. And when we looked at the nuclear entry efficiency and integration efficiency by measuring reverse transcription and nuclear entry and infectivity, what you see here is quite significant. For example, you see infectivity is reduced in CPA depleted cells. There is reduction in reverse transcription and nuclear entry, but the level of integration reduction is much more higher. And if you take nuclear entry efficiency, which is essentially the level of reverse transcription over level of nuclear entry, what you see is cyclophilin A depletion has limited impact of the virus to enter the nucleus. However, the integration efficiency is dramatically reduced, providing further support that a capsid binding host factor, in this case cyclophilin A or CPA, regulates the ability of the viral DNA to efficiently integrate into the target cell chromosomes. And this, in addition to these three different approaches, we also, as I indicated, study the pre-integration complex or the PIC. Hopi, can you mute yourself? Waldik, thank you. And let me go here. And the pre-integration complex, or PIC, which is formed in an infected cell after the RNA is converted into a viral DNA copy, has been a great tool to study the mechanism of HIV integration. In fact, it was the studies of the PICs that led to the development of raltegravir. So the pre-integration complex, or PIC, is composed of viral and host proteins. It protects the viral DNA from cellular sensors. It carries out viral DNA integration in vivo or in infected cells. And one of the fundamental things is these integration complexes can be extracted from infected cells, and they retain the viral DNA integration activity in vitro. For example, if you take a substrate, viral, not viral, any DNA substrate, and add to the pre-integration complex, the viral DNA will get inserted into it, and you can measure the viral DNA integration activity. And we have been using this assay for a number of years, in fact, more than 10 years now in the lab. And I'm not providing all the details of the biochemical analysis, but what we do is we take the pre-integration complexes from the infected cell, provide a 2 kilobase double-stranded DNA, and then we measure integrated viral DNA levels into this by qPCR. And here is the representative data. There is the copies of integration we measure, and this is specific because if we add an integrase inhibitor, in this case raltegravir, you can see there is a dramatic reduction in viral DNA integration copies. And we have published a number of papers in this area, and we'll be happy to talk to anybody who is interested in PIC assays. But the point here is not only we can extract these PICs, we can biochemically analyze them to study their function and study the function of different factors in these complexes. For example, here I'm showing you the PICs that are fractionated on a sucrose gradient, what you see is that the fractions that contain the integration activity also contains the maximum level of viral DNA, because viral DNA is essential for integration. But what we reported for the first time to the field is that the functional pre-integration complexes also contain a measurable level of capsid. And this was reported in 2019, and most of the reports that an intact or near intact capsid enters in the nucleus was mostly from publications after 2019. We believe that our observation had a significant impact on the development of new model that some versions of intact capsid goes into the nucleus. But the point here is we have this biochemical tool that allows us to study capsid's role. For example, if we take the pre-integration complexes and add a capsid inhibitor, what you see is that by treatment of these PICs with a capsid inhibitor inhibits integration activity quite robustly. And not only that, if you use a capsid inhibitor, in this case PF74, the level of capsid, this is again a fractionation assay where I'm showing you the capsid levels in these fractions that have integration activity. What you see is the presence of capsid inhibitor reduces the level of capsid in these functional PICs, which are shown in a bar graph here, suggesting that certain level of capsid is essential for the viral DNA integration activity by the pre-integration complex. In addition to that, now we can take these pre-integration complexes and assay for integration activity using capsid binding host factors, in this case CypA. If we add cyclophilin A or CypA to the PICs, you can see there is a dose-dependent increase in integration activity. And this activity is specific because if we add a cyclophilin A inhibitor, which is cyclosporine A, you can see the stimulating effect of CypA can dramatically be reduced, suggesting that CypA's effect on the integration activity is dependent on its binding to capsid. So the bottom line here is we believe utilizing the pre-integration complexes to study the role of CA in viral DNA integration is a great tool. So just to summarize, our studies clearly demonstrates that capsid binding inhibitor, in this case PF74, inhibits a nuclear event, HIV integration, and HIV capsid regulates PIC-mediated integration activity. CA mutants, both laboratory adapted, which I didn't show you data, and clinically relevant viruses are defective in HIV DNA integration, and CA binding host factors such as CypA promotes HIV DNA integration, providing further support to our hypothesis that capsid plays a direct and functional role in the nucleus of an infected cell. So in addition to capsid being a multifunctional role, which has been reported by many laboratories to promote reverse transcription, to target host, serving as a target for host restriction, for role in nuclear entry and protecting the viral DNA, we have been able to demonstrate that CA controls viral DNA integration. And why this is critical, but before I say why this is important, we are currently working on a couple of models to understand how the capsid protein regulates DNA integration. We believe the capsid protein is essential for maintaining the quality of reverse transcription, not the quantity per se, because viral DNA integration is dependent on 3' end processing. It is also important to maintain PIC composition. If the PIC is altered from its composition point of view, what host factors are there or not, then it cannot carry out viral DNA integration. We also have some data that suggest that CA helps in viral DNA integration by directly regulating how the PIC interacts with the human chromosomes. So stay tuned. We have some great data to test or confirm these hypotheses. But why these studies are significant? Because, as you know, CA targeting inhibitors are being tested for as the ultra potent drugs. Therefore, understanding how CA regulates HIV infection is critical for developing superior CA targeting drugs. Just remind, the lenacapavir is not utilized for as a frontline drug. It is still restricted for specific HIV infected individuals. And we believe our studies of CA mutants that escape CTL mutants are critical for developing strategies to use CTLs to naturally control HIV infection. As many of you know, ARTs are not curative, and we have to develop alternative strategies where HIV infection can be controlled naturally. So with that, there are a lot of people involved in these studies. Our lab past members, many of them have moved on to different fields or different institutions. The current lab members, we collaborate heavily with the Balasubramaniam lab, who paved the way for these early studies and now has his own lab at Meharry. You can see we collaborate with many leaders in the field throughout the United States. And I'm not going to read everybody's name, but we utilize complementary approaches from many laboratories, a number of labs in Meharry, and none of these studies will be feasible without funding support from NIH. Specifically, the support from Tennessee CFAR and RCMI has been hugely instrumental for our research. With that, it's 3:45, and I'll be happy to answer any questions or comments. Thank you very much.